So now let's take a look at the small intestine and the large intestine. So we'll start with the small intestine, which is made up of three major regions. The first region is called the duodenum. Second region is the jejunum, in purple here, and then the last region is the ileum, which will connect to the cecum of the large intestine. You can see the duodenum connects to the stomach, as well as the pancreas, the liver, and the gallbladder. Remember, we talked about that ampulla that allows those secretions from those three glands to empty into the duodenum as well. So the small intestine is where most of the chemical digestion and most of the absorption process will be happening throughout the digestive tract. And it's referred to as the small intestine, not because it's shorter than the large intestine, but because the diameter is smaller than the diameter of the large intestine. So the small intestine is actually longer than the large intestine. Now, looking at the duodenum, specifically the first part of the small intestine, we're going to see some mucus being produced in the small intestine, and that mucus is there to protect the mucosal membrane from the stomach acid, the hydrochloric acid that is mixed in that chyme that's coming out of the stomach. We also know that we're going to have bile being secreted into the duodenum, which will help emulsify fats. We have those pancreatic enzymes that will be secreted into the duodenum, which will help with the chemical digestive process of many of those food groups that we have eaten. We're also going to have pepsin coming from the stomach. It will be inactivated, though, because it's going to have played its role already in the stomach. It will be inactivated by that elevated pH of the duodenum, thanks to that mucus and that alkaline-rich juice that's being secreted by the pancreas into that section of the small intestine. Moving to the jejunum next, that's the middle region of the small intestine. This is where most of the digestion, most of the chemical digestion and absorption process is actually going to occur. So it makes up about the first 40% of the small intestine past the duodenum. And then the last 60% of the small intestine is made up of the ileum. In the ileum, we're not going to have a ton of digestion occurring, but we will have some more absorption occurring of those nutrients that we have digested, mainly in the jejunum. Now the ileum is going to join up with the cecum at what we call the ileocecal junction, and at that ileocecal junction we also have an ileocecal valve, which can regulate food passage from the small intestine into the large intestine. Now, looking at the internal anatomy of the small intestine, we will see that there are three inner folds or projections that are there to help increase the surface area of the small intestine for more nutrient absorption. So the first type of projection that we see is inside of the small intestine. If you were to look inside of it, it would look wrinkly. It would have these little wave-like projections in it called the circular folds. So these are foldings of tissue inside of the small intestine. And then if you were to zoom in on those circular folds, you would see what are called villi, which are these guys here. They look like little finger-like projections projecting up off of those circular folds. You can kind of see those folds a little bit in this diagram. And then if you zoom in on a singular fold, you will see these little finger-like projections on that fold known as villi. Now the villi, if you were to zoom in on them, you can see individual columnar cells, simple columnar cells, which contain microvilli. These are little teeny tiny hair-like or brush-like cell surface extensions on the surface of those columnar cells, which are also there to increase surface area. So all three of these, the circular folds, the villi, and the microvilli, are there to increase surface area of the small intestine for better absorption of nutrients. We also know that these microvilli create what we call a brush border on the surface of these cells, and that brush border helps form brush border enzymes. It contains brush border enzymes, which are useful in helping us digest certain food groups in the small intestine. So now moving to the movements that occur within the small intestine, contractions in the small intestine. There are two different types of contractions that we see in the small intestine. We have what's called segmentation, which you can see over here, and then we have peristalsis, which you can see here on the right. Now segmentation, the main purpose of this type of movement is to churn the contents within the small intestine. So why do we want to churn these digestive contents? Well, we want to churn that material with the enzymes that are being produced by the brush border. We also want to churn it with the enzymes being produced by the pancreas, We want to mix that chyme with the juices, with the bile that's being secreted by the gallbladder and the liver. And so if we mix those contents, better, we are increasing the surface area of that food material to be able to come into contact with those digestive enzymes, with that bile, to better digest chemically those components that are found within that food. So it's going to churn those materials together, allow them to mix with those digestive enzymes, ensuring that they're coming in contact not only with the digestive enzymes, but also with the absorptive cells of that small intestine lining, so that we can absorb as many nutrients as possible as well. Now, the peristalsis movement is allowed for by contractions, like wave-like contractions of the stomach. Of the smooth muscle, right, of the lining of the small intestine. And when that circular and longitudinal layer contract, they form this wave-like contraction to push the material in one direction towards the colon. So this is going to help move that chyme, move that food residue towards the colon. And as we're moving it in that direction, we're also going to be performing segmentation to churn that food up. And so you'll see that it becomes better and better mixed as it moves through the small intestine towards the colon. So now we're going to take it to the large intestine. And you can see here this ileocecal junction right here. So here's the ileum coming in, and this is where it joins up with what we call the cecum. The cecum is the first part of the large intestine. So the cecum is this pouch-like attachment here. And then hanging off the cecum, we see what's called the appendix. This is about the size of a pinky finger. And the appendix is going to contain a lot of lymphatic tissue. There's lots and lots of lymphocytes in it. Sometimes bacteria can aggregate in that appendix. It can set up inflammation because of that, and that can lead to appendicitis. Appendicitis can be very dangerous because if the appendix ruptures, there are trillions of bacterial cells found in the large intestine and in the appendix that could leak out into our body cavities and end up causing sepsis. So appendicitis would end up leading to having the appendix removed to prevent that from happening. So again, we have the cecum, this pouch-like beginning of the large intestine. And then from the cecum, we move up into what's called the colon. So the colon actually has 4 regions to it. We have what's called the ascending colon, which travels up. We have the transverse colon, which travels across. The descending colon traveling down here. And then we have what's called the sigmoid colon, which forms this comma shape at the lower region of the large intestine. Now from the sigmoid colon, we then enter into what's called the rectum. Right? The rectum is the region of the large intestine that when feces enters into the rectum, it will kind of stimulate that defecation process and make us want to go to the bathroom. Below the rectum, we then have the anal canal, which empties out of the anus. So those are the major regions, okay, of the large intestine. You'll also notice that there are 3 bands of muscle, okay, called teniae coli. So these are longitudinal strips of muscle. There are 3 of them found on the outer layers of the large intestine. And these strips of muscle are actually a lot tighter than the tissue around it. And so that ends up causing the tissue around it to be pulled in, and it causes it to pucker out into these pouch-like structures called haustra. So these haustra can perform haustral contractions to help move the material more quickly in these very large movements from one end of the large intestine to the next. Now down here, once the waste material, okay, makes it from the cecum, from the colon, finally into the rectum, we then have 2 sphincters in the anus. We have what's called an internal anal sphincter and an external anal sphincter. The internal anal sphincter is made of smooth muscle, and it will relax when the feces enters into the rectum and stimulates that defecation reflex. But the external anal sphincter is made of skeletal muscle, which means that we have conscious control over it and can keep it closed long enough for us to be able to make it to the bathroom. Now, in this large intestine, we also have what's called our microbiome. So this is a community of microbes or bacteria that we find in the gut. There are over 1,000 different species in the large intestine, and they contribute about 10 pounds of body weight to our body. So when you defecate, when you remove feces from the body, a large portion of that feces is actually made of bacteria. About 30% of that feces is made of bacteria. And those bacteria serve some useful purposes for us. They help us synthesize certain types of vitamins, like vitamin B and vitamin K. And they do this by breaking down complex carbohydrates that we cannot digest on our own. So for instance, like cellulose. Cellulose is a complex carbohydrate that we cannot digest. These bacteria help break those complex carbohydrates down, and they produce beneficial vitamins for us when they do that. Now, along with producing those beneficial vitamins, sometimes these bacteria also produce gas, or flatus, when they break down these complex carbohydrates. And so that can lead to gas in the large intestine. It can also lead to a really bad smell as well, because sometimes that gas contains noxious odors that are coming from the byproducts produced by those bacteria. So the feces in the large intestine takes about 36 to 48 hours to produce feces. So the residue is going to stay in the colon, the transverse colon primarily here, for about 24 hours before it passes into the descending and sigmoid colon and finally into the rectum. And that feces is made up of mostly water. 75% of the feces will be made of water, but most of the water is actually absorbed by the large intestine during its route to the rectum. About 30% of the feces is made of bacteria, about 30% is made of undigested fiber, complex carbohydrates that we cannot digest, and then about 10 to 20% of that feces is fat. So that feces moves through large intestine through what we call haustral contractions, which is going to help continue to mix and churn that residue, but it's also going to promote water reabsorption from the residue and salt reabsorption from the residue that we might want to hold on to. Those mass movements are very strong contractions, and they only occur a few times a day, meaning that these movements are going to be very large movements. Again, that defecation reflex is going to be set off whenever the feces enters into the rectum here and causes it to stretch. That will cause that internal anal sphincter to start to open up or relax, and that will trigger our urge to defecate, where we will then relax the external anal sphincter to allow that fecal matter to leave the body.